Good afternoon, everybody. Welcome to the third and final day of the UK Spine Annual Conference. My name is David Bowcott. I am the Partnerships Manager for UK Spine, and I have the pleasure of chairing the first two sessions this afternoon. I would encourage you all to get involved with the conference as much as you can using the networking tools provided by Hopin. And if you have any questions about how to do that, please click on the Sessions tab on the left-hand side of your screen. This will take you to the help desk, and a member of the UK Spine team will be there to help you. So on Wednesday, we focused on biomarkers of aging. We asked what it means to age well, and we discussed how we can practically measure that. Yesterday, we looked at new medicines. We talked about how we can work together to make new drugs that will improve health in old age. And today, for the first session, we turn our attention to existing drugs. And we want to know how we can best use those to support healthy aging. Now, to discuss this, we're very lucky to be joined by Graham Russell, Emeritus Professor of Musculoskeletal Pharmacology at the Universities of Sheffield and Oxford, and also James Edwards, who leads the Musculoskeletal Aging Group at the Nuffield Department of Orthopaedics, Rheumatology and Musculoskeletal Sciences at Oxford. Graham and James will be talking about bisphosphonates. Now, this is a class of drugs with a long history of clinical use, and we hope a lot of exciting potential in the future for promoting healthy aging. So I can welcome Graham Russell to the stage. Graham, if you can turn on your camera and microphone, please, and we'll begin the first presentation. Hello, Graham, welcome. Your slides will be brought up now, and I will let you get on with speaking.
Fine. Well, many thanks for running this session. We're passionate about bisphosphonates and do believe that they have enormous potential in the area that we've been discussing over the last few days. Cheap and cheerful and very effective, I would summarize them as. So this is where I live. So if we can up and down the M1. But if we can move to the next slide, I'm having to have the slides controlled because we had a slight hitch. So this asks the question of what are bisphosphonates. I imagine there may be some folk who don't know. They're actually structural analogues of pyrophosphate, which is a naturally occurring substance in the extracellular environment. It acts as an inhibitor of calcification and is therefore nature's water softener. And inside cells, it's a common byproduct of reactions involved in biosynthesis, such as cleavage of ATP and amino acid synthesis, fatty acid synthesis, and so on. By virtue of the two phosphate groups joined by a carbon, the bisphosphonate is different because it's chemically stable. And these are the things that are used in medicine. Because of the carbon and the two phosphates, you can put a whole variety of different substituents onto the center carbon. And the two phosphate residues gives it strong calcium binding properties and therefore an affinity for bone mineral. And the serendipitous finding that we made around 50 years ago was that they not only inhibit calcification, as you might have predicted from the pyrophosphate history, but they actually inhibit bone resorption, which is actually why they're used so much in medicine. We now know a lot about how they work, and they still are the major drugs used in osteoporosis and bone oncology. So if we just go to the next one, just to say that we actually had a birthday party for the 50th anniversary of the discovery of bisphosphonates, reported in Nature and Science 1969, and this was held in Sheffield, as you can see there. The proceedings are being published in the journal Bone, and if anybody wants reference for that, we can provide it. Lots of review articles, up-to-date review articles appearing. Okay, the next one. So this is a pictorial history making a number of points. So it's a 50-year journey from the water softening era right through to the current time. And the pyrophosphate, acting as a water softener type agent, did see commercial life added to toothpaste, like, for example, Crest toothpaste, much used in the dental area, but also as an additive to water softeners before phosphates became unfashionable. This work actually took place, the discovery of the bisphosphonates took place in Davos, Switzerland in the late 1960s. And I was working with Herbie Fleisch, who's sadly no longer with us. And the bisphosphonates that we worked on were supplied by Dave Francis from the Procter & Gamble company. Now, it took a long time to work out exactly how they work. And it was actually working in the SGC with colleagues in the SGC group in Oxford that you heard about yesterday, that we were able to elucidate the structure of this enzyme in the mevalonate pathway, which they inhibit. There's a recent review of the pharmacology cited at the bottom there, but again, if anybody wants references, we can provide them. It's become a huge field. The last count of over 33,000 publications cited in PubMed alone. So if we move to the next slide, please. So if you ever get a prescription, and a lot of the population do, for your osteoporosis, you may get a package like this, although these days these drugs are generic and don't come with the branded packages in the era when the pharma industry looked after them. They were drugs marketed by Merck and Roche and other big pharma companies, Novartis and so on. Next slide. So some of the key properties are and historic effects are shown here. So I've already said they bind strongly to bone mineral, and they actually enter the osteoclast, the bone resorbing cells. And you can see there some beautiful pictures using fluorescent bisphosphonates actually going into an osteoclast on a bone surface. They have great tissue selectivity. So when we start to think about non-skeletal effects, we've got to also think about how much gets to the tissues outside the skeleton that might be a benefit in some of the multimorbidity conditions. One of the first uses of bisphosphonates was actually as bone scanning agents. If you link them to technetium-99, take these images, Ignace Fogelman, who led nuclear medicine at Guy's Hospital until very recently, sadly also no longer with us, pioneered in this area. And these scanning, use in this way as scanning agents goes on to this day. So that application has survived 50 plus years. Next one, please. So the diseases of excessive bone resorption, which they're ideally suited for, some of the major ones are listed here: Paget's disease, myeloma, bone metastases, and osteoporosis. And I'll give you a couple of examples of each of these, more or less. So the next slide actually shows you that the first applications in Paget's disease were done in Oxford and published exactly 50 years ago. Roger Smith, who's still with us, we did studied these first 5 patients, and 4 of them actually normalized their bone turnover, being given atidronate, which was one of the early bisphosphonates, orally, and it was very effective, as you can see. That's the old Nuffield Orthopedic Centre, consisting of huts. These days they have buildings. Okay, next slide, please. So this is just capturing that same concept again. These are the bisphosphonates that all became blockbuster drugs, billion-dollar sales plus, for the treatment of osteoporosis. And the aim is to prevent fractures, wrist fractures, spine fractures, but particularly hip fractures. And you will see on the right there zeledronate, which sort of stands out, and you'll hear a lot about this drug, because it's the one that's given IV only once a year. So if we're thinking about giving a bisphosphonate along with your flu jab every year to stop everything else nasty happening to you, this would be the one we'll go for. Next, please. And this remarkably long duration of action of this drug is sort of shown here. For bone, we actually have wonderful biomarkers. We have a fragment of collagen as a measure of bone resorption, and a propeptide of collagen as a marker of bone formation. And both of these remain suppressed after a single injection of 5 milligrams of seledronate. A single infusion has an effect that lasts for up to 5 years, which is truly remarkable. Next, please. Now, bisphosphonates are not without competition, but the competition, in my view, has been very exciting, has come, and then is now undergoing a bit of difficulty. So the big competitor is an antibody to rank ligand, which is the major cytokine that drives osteoclast differentiation. And these antibodies also block bone resorption, used in cancer and osteoporosis. The one problem with them is that this antibody has to be injected twice a year. And if you miss injections, you get a very rapid rebound of the bone resorption. And that's beginning to become quite a clinical problem. This contrasts with the bisphosphonates, which, as I've said already, have that long duration of action. Next, please. So if you look at the sort of overall picture of all the drugs that are being used to prevent fractures, you see a whole list of them here, and the 4 bisphosphonates are shown in green. 3 of them reduce hip fractures, which is really the fracture that one really wants to reduce because of the huge costs associated with that and the morbidity also associated with it. Denosumab does the same. Adanacatab, which is a cathepsin K inhibitor, also did the same, but was never approved as a drug because of some side effects, actually demonstrating the challenges of drug development, even after spending millions of dollars on the development. Okay, the next one, please. So the other big application area, apart from osteoporosis, is in cancer, where the aim is to prevent skeletal-related events, like hypercalcemia, bone loss, fractures, loss of height, and all those things. And you see the type of lesions you can see on X-ray in somebody with myeloma or breast cancer. The first bisphosphonate used in this area was pomidronate, and then it was followed and more or less replaced by seledronate, a Novartis drug. Next, if you just press again. Yeah, and actually what's been shown in cancer studies is that the bisphosphonates prolong the survival of patients who get them under certain conditions in breast cancer and also in multiple myeloma. So they have this mortality benefit even in the cancer setting. Next slide, please. Now, understanding how these drugs work is really important, but we didn't know until more than 20 years of use what the exact biochemical mechanisms were. We thought originally it was all to do with the physical chemistry of bone mineral. And the reason why this is important, if you just press again, please, is that not only does it help understanding how they work on bone resorption, but it takes us a first step in explaining how we may see some non-skeletal benefits or effects because of the pathways that get affected. So the next slide actually shows this. So for this group of bisphosphonates you see on the left, they get incorporated into ATP by a back reaction of tRNA synthases. Very interesting biochemistry. And these mess up mitochondrial function and other things and may cause apoptosis in cells. On the right, the ones that are probably more interesting in the context of this meeting are the nitrogen-containing bisphosphonates, which block to inhibit prenylation of a multitude of GT-binding signaling proteins inside cells. And the way they do this is shown on the next slide. Here, you can see that they actually act as specific inhibitors of an enzyme in the mevalonate pathway below the step at which statins work, and they block the production of these lipids that attach to the GT binding proteins. So by inhibiting a single enzyme, you can actually interfere with the function of probably 100 or more signaling pathways within cells. And that's the key to their action, and it's the key to the non-skeletal effects as well. Next slide, please. So here are the formulas. There's a statin up on the left there, and you see a couple of the bisphosphonates on the right. And it also raises two additional questions. One is statins combined with bisphosphonates might be expected to have additive effects under some conditions because they inhibit in the same pathway. And there is a question of whether statins can do anything to bone and whether bisphosphonates can do anything to lipids. And for the most part, the crossover is really small or non-existent because of the tissue selectivity. The next slide, please. So if we partition our group of bisphosphonates, which we have around, all of these have been used clinically in some settings, we divide them into these two groups. The first group at the top there don't have nitrogen in them. Those are the ones that go to ATP analogs. And then the bottom two groups, the older and then the newer ones at the bottom here. The bottom group are all designed by medicinal chemists to optimize the structure activity. And we on the bottom right here have a compound called OX14, which James Edwards, who follows me, has included in some of his studies, just showing that one can go on innovating with bisphosphonates and produce even better ones. Next, please. So this is how we summarize how bisphosphonates work and how we actually rank them in potency on different effects. They have different effects on bone mineral binding. There's a ranking order, and same in terms of inhibition of FPPS, with the most potent actually being nanomolar inhibitors of this enzyme. Next, please. So this is the pathway in a little more detail. So you can see the chemical structures, the crystal structures of two of the enzymes in the pathway that bisphosphonates affect. But there's a little bit of evidence that they may act at some of the other sites as well. But again, this comes back to the liver and bone effects, which seem to be somewhat distinctive. And as I say, the important thing about the bisphosphonates is blocking a prenylation of GTB binding proteins. Next, please. So what do we have in the way of studies of effects beyond these bone applications? Well, in the last 10 plus years, there have been a number of observations that suggest that they may have a lot of other good properties in terms of human health. They actually extend lifespan. This has been shown in a number of studies. They have cardiovascular beneficial effects. They, in certain situations, seem to prevent cancer. And there are even effects on protozoa and parasites demonstrated experimentally. We also have an interest in using them for drug delivery to bone, which is not a topic for today. But the next slide I put in really to dial back to what Ferguson was telling us yesterday about his group's interest in protozoa and parasites. Interestingly, bisphosphonates inhibit a whole variety of protozoa and parasites, probably by inhibiting in the same pathways. But there's not enough work done yet to translate this into clinical utility. It's a neglected area and really ripe for study and application. The next, please. So let's focus in on the lifespan, health span, and mortality questions and go to the next slide. This was really started from a trial done by Ken Lyles at Duke University in the US, who was studying quite sick patients, old patients who were getting hip fractures and giving zoledronate to see whether you could prevent further fractures. So this is your typical patient with a hip fracture, comes into hospital. Can you prevent them when they go out, if they go out, from getting another fracture? And they observed quite unexpectedly that the mortality of these patients was lifespan was actually extended by around 28% in the people who were given the zoledronate. A really unexpected finding, which set off a whole slew of work to try to understand how this came about. Next, please. So the overall reduction was 28%, as I've said, in this particular study. Next, please. So again, just to make the point, this was an unexpected observation, which really opened up the idea of using drugs like zoledronate for these other good effects on human health. And interestingly, when Ken and his colleagues started looking at why there was a reduction in mortality, they found, among other things, fewer pneumonias and actually cardiovascular events in these elderly patients. And we'll come back to that topic in just a minute, because we heard about the pneumonia story yesterday. Next, please. Now, there's been another study involving about 2,000 patients, 1,000 controls and 1,000 put on zoledronate from Ian Reid in New Zealand, where they've given a bisphosphonate infusion not yearly, but every 18 months to people with low bone mass who hadn't yet had fractures. And in this study, they also found an almost significant reduction in deaths in the people who got the zoledronate during this 6-year study. It's a beautiful study in the New England Journal of Medicine 3 years ago now. The next, please. So 35% reduction. Only 2,000 patients overall in this study. So it does show that you can show some of these benefits in really quite small trials. And I think this is a message as we discuss logistics of what we might do with the sort of data that we already have. The next, please. This is very difficult to read, and I apologize for this, but this is actually the table from that paper showing not only the reduction in deaths, but the reduction from myocardial infarction, other cardiovascular outcomes, and actually a significant effect on reducing cancers. So all these hints, or more than hints, that there are other benefits. Next, please. So where does one go with this? Well, there are a number of pointers and lots of further studies out there that sort of help us to understand what is going on. One of these is Hutchinson-Gilford progeria syndrome, which is a genetic disease of premature aging, and the progeria syndromes. And actually statins plus bisphosphonates are being used in that, in patients with that. Difficult studies to do because of the difficulty of measuring outcomes. But these, apparently these patients are not doing worse, let's put it that way. The next slide actually shows an animal model for that disease. This is a prenylation defect disease. And you will see that mice, that you can actually make genetically modified mice with this syndrome. And next, please. This is a study that actually shows that if you give a mixture of statins plus bisphosphonates, you can extend the lifespan of these mice by 2-fold. Next, please. So if we just list the growing number of areas in which we might expect to see effects with bisphosphonates, they're listed here together with a few of the references: the mortality, lifespan, cancers, heart attacks, survival in intensive care units, and survival after pneumonias. A very interesting paper from Singh and his colleagues just published. Next, please. So I'm just going to show you a couple of these examples. This was a huge study of nearly 20,000 patients with rheumatoid arthritis who have an increased level of cardiac risk, and those on bisphosphonates had a lower mortality rate. Next. as you can see, 28% reduction in heart attack. Next. And this is the very interesting intensive care unit survival study from Sydney in Australia. And again, if you just click on that, there's a 59% reduction in in-hospital mortality in patients who had a bisphosphonate when they came into an ICU unit, regardless of the reason why they were admitted. It's a truly stunning result. Next. So what are the underlying mechanisms that we might think about here? We'll start with the next slide to see how this was sort of taken further and what the potential mechanisms might be. There's a whole list here of things that might be happening. Things coming out of bone like toxic metals or growth factors, effects on lipids. Bisphosphonates have weak statin-like effects on calcification of blood vessels, immune effects on gamma delta T cells, stem cell aging, cell senescence, autophagy, protection from radiation, and so on. And James will be telling you about some of these in the experimental work that he's currently doing. The next. This was a study that's not yet published, but was run by Ken Lyles, where they took pigs and infarcted them, gave them heart infarcts, and showed that if you gave them zoledronate, they actually recovered their heart function significantly. Ejection fraction is a measure of that if they have received zoledronate. So experimentally, you can actually demonstrate this benefit after heart attacks in pigs. The next. And we actually took this data to the FDA a number of years ago, who were extremely impressed by it and encouraged a trial to see whether you could work on people who had myocardial infarcts in hospital and see whether you could improve their survival by giving a bisphosphonate. The trial would have been very costly. It was going to be run in the US. And despite walking around Wall Street, we didn't find the funding. So that's one open for further development for anybody who would like to pursue it. Next, please. Next, please. So just a couple of things towards the end here. We actually started some studies trying to understand the effects at a cellular level, which is the sort of area that James is now following up on. Next, please. So this was a paper that was published now about 5 years ago, showing that you could extend stem cell survival and protect them from radiation damage. And that was seen with zoledronate in these particular studies. So interesting ideas in relation to tissue repair. Next, please. And then something that's not actually been fully published yet, effects on gut regeneration. And this is another irradiation situation. And the next slide shows the pictures from this study where if you irradiate animals, their guts, that you affect the stem cell population in the gut. But if you give them zoledronate, you actually stop or prevent that type of radiation damage. Again, really interesting ideas. Next. So there are effects on the immune system of drugs like zoledronate, which have been known for a long time. And this is actually the biochemical mechanism by which they're mediated. If you block the FPP synthase, you put a dam in the way of the pathway and accumulate this upstream metabolite called isopentenyl diphosphate, which acts on gamma delta T cells and causes their proliferation and cytokine release, which can cause fever in some patients, which is part of the innate immune system, and affecting gamma delta T cells in this way has all sorts of potential applications, including in pulmonary infections. The next. So I was very intrigued by what John told us yesterday, which we'd had a preview of, John Overington, about the influenza story. where he talked about, among many drugs looked at in terms of their impact on influenza, 3 came out: metformin, PPIs, proton pump inhibitors, and bisphosphonates as potentially having effects. And the story about preventing pulmonary infections is a recurring one. The next. It even leads us to think that there might be applications in the COVID situation, in terms of either affecting the severity of the lung inflammation and the subsequent tissue damage and fibrosis. Again, there's literature out there to suggest that that's possible, although we haven't yet, I think, seen any clinical data on whether patients on bisphosphonates fare differently, or even on statins fare differently from those not on those drugs. Again, it would be another disease area to study properly. The next, please. So this is my attempt to summarize sort of kind of where we are and make the case for bisphosphonates as candidates to prevent multi-morbidity. You know, these drugs have been around for 50 years. They're safe and effective. We know a lot about how they work. We've got these 2 studies showing effects on mortality and effects on reducing pneumonias and cardiovascular outcomes, even in quite small size trials. And many observational studies on larger numbers show similar sorts of effects in terms of cardiovascular and pulmonary infection effects. The non-skeletal effects are under study, and James can tell you about some of the interesting recent advances in this area, which demonstrate plausible mechanisms for how this come about. Finally, bisphosphonates are generic, inexpensive, and you could treat somebody theoretically for less than £10 a year. So cost is not an issue. The next. This is the final one. This really makes the case for zoledronate as being the one that one might prefer to use among all the others, because one gives it parenterally. You don't have the problems of compliance with oral medication. And we've got these various studies we've been talking about where the effects have been seen. And again, it's a very effective drug. The one big drawback is that at the moment it has to be infused. So looking for other ways of giving it, either by oral administration or transdermal, would definitely be something worth aiming for, not only for standard indications, but also if it's going to be added to multimorbidity studies. With that, I finish, and thank you very much for your attention. Thank you.
Thank you very much, Graham. That was a really sort of fascinating story of a drug that started in clinical use without us understanding how their mechanism of action worked. And we're learning more and more about the different effects and benefits they may have every day. So I'd now like to invite James to turn his camera on and turn on his microphone. And James is going to tell us a bit of the work he's been doing in his lab that goes on to investigate the pathways by which the non-skeletal effects of bisphosphonates appear. So James, over to you. Thank you very much.
Thank you, David. Okay, I'm sharing my screen. I'm hoping you can all see this and see me. Do shout out if you can't. So thank you, yes, to the UK Spine team for asking us to talk here, but also for having the boldness to help fund some of our preliminary work in helping you understand the potential for bisphosphonates. So Graham's done a great job in describing how he and his colleagues identified and brought bisphosphonates from its initial discovery as something that binds calcium to one of the biggest drugs that are used in the world today, specifically in the field of accelerated bone disease. But more excitingly, what we now might be able to find out about what bisphosphonates can do beyond bone, and that's really where we've been focused. So I'm going to, slightly opposite what Graham's done, I'm probably going to throw too much data your way in the short time that we have. But what I want to try and get across is that there are many other things that bisphosphonates might be able to do beyond just binding bone. So very briefly, where I sit is about where that question mark is. For the past 15 years in running the Aging and Regeneration group, we've been particularly interested in this inextricable link between getting older and developing aging-related disorders. This has been focused primarily on the musculoskeletal system, but has more recently expanded into a number of other aging-related disorders. And trying to see whether there are common factors which exist, which control aging and longevity, and also normal cellular and tissue function. And we're specifically looking to try and identify new aging-related mechanisms, to characterize both old and new aging mechanisms, and specifically to try and target these to straighten, to square the curve that Tom Jackson spoke about in the first talk of this meeting. And not only to try and increase our health span, but possibly try and increase our lifespan as well. And that, of course, is where we come up against bisphosphonates. So I'm not going to dwell on this too much. Graham's already done a really good job of summarizing all the literature that has highlighted that bisphosphonates are associated with a number of other beneficial effects, which go way beyond just improved skeleton. And these may be mediated by the fact that, you know, the patients are walking around a little bit more with slightly stronger skeletal integrity, but also possibly by direct effects of bisphosphonate within these areas themselves. So, as Graham mentioned, bisphosphonates were originally shown as a calcium binding agent, and in many ways, I think this really interesting effect of these cells has become a little bit of a curse in their expanded use beyond bone, because they go to bone, they bind to bone quite avidly. They get internalized by osteoclasts when osteoclasts identify areas of damaged or defective bone for removal. So osteoclasts get exposed to quite a high level of bisphosphonates when they do this. But it's led to the impression that bisphosphonates are only a bone drug, only exist within the skeleton, and only affect cells which are able to endocytose them. So when you think about this panel of cells that I'm showing on the right that might be impacted and might be having an effect on some of these disorders that Graham's talked about, it does make you ponder how bisphosphonates might be functioning there. So this was the challenge that we tried to take up with this UK spine project. So we wanted to do a study, which was essentially a cellular screening study, where we will look at multiple cell types using multiple different bisphosphonates, multiple doses, multiple time points, and look at a number of different outputs as well, including normal cell biology approaches, but also approaches which we know are altered during aging, such as changes in oxidative stress and DNA damage. And a large part of this work was done by Helen Knowles and Jinsen Louis, who I'll mention as we go through. We've coupled this with another project, which we were lucky enough to work with Ian Reid over in Auckland, who Graham indicated a little bit earlier, who conducted one of the most convincing studies that bisphosphonate does have multimorbidity effects. And this was published in the New England Journal of Medicine. We were able to get plasma samples from these patients who were on this study and screen them using this expensive new screening approach, proteomic approach, which is done by the Somologic firm, where we get 5,000 different readouts for each sample. So what we've got is a cellular biology theme looking at effects of bisphosphonates, as well as a human secretome impact of bisphosphonates too. So if we firstly think about bisphosphonate distribution and where bisphosphonates are, we know they go to bone, and this is what these images are showing by our colleague Shelley Lawson up in Sheffield. So this is using drugs that were made similar to those made by Hal Ebertino, where bisphosphonates are tagged with fluorescent probes. Using a mouse, you can see the red bisphosphonate in the tibia and in the vertebral body binding to areas of calcified bone. But we also see fluorescent tagged bisphosphonates taken up by macrophages, and this is what's been shown in the images on the top, as well as by monocytic cells, which is shown in the image on the bottom left, in addition to other cells which you wouldn't really expect to be able to take up bisphosphonates if this phagocytic or endocytosic process is crucial, such as osteoblasts and breast cancer cells. These little green dots being the fluorescent bisphosphonates. And this was the work of Mike Rogers and Fraser Coxon. In addition, as Graham mentioned, bisphosphonates have also been shown to activate the group of T cells known as gamma delta T cells. And this was shown nearly 20 years ago. So through this mechanism, there's the potential for bisphosphonates to impact a number of other systems, such as having anti-cancer properties, or interestingly, having antiviral properties as well. So what we wanted to do was to try and, as I said, try and confirm what bisphosphonates might be doing in these tissue types. So taking a panel of these drugs, as well as a panel of these cell lines, we've been treating bisphosphonates at different doses, and this is the work of Helen. So as you can see here, at high doses, these are the graphs that are shown across the top. We've got 100 micromolar showing pretty much apoptosis in all of the different bisphosphonates that we used, and this is in the heat maps at the bottom. You can see across all the cells. So at these high doses, we do see apoptosis, specifically with the nitrogen-containing bisphosphonates. But if we dial the dose of bisphosphonates right down, and this is going down 10,000, 100,000 levels to picomolar concentrations, and again expose our same sort of cells to these, what we see, specifically in the heat maps at the bottom, is an overall creep towards green as we move from left to right. As the dosing is lowered and the days increased, we get an overall increase in cell growth when cells are exposed to low doses of bisphosphonate. And this was particularly striking in the cardiomyoblasts, where again, 100,000-fold less than what you might see to induce apoptosis, down into the picomolar range, actually increased cell growth by over 600%. Now, we've heard a little bit about the whole hallmarks of aging from others during this meeting, and of course this has highlighted a number of different processes which we know to be impacting human aging, such as telomere shortening and mitochondrial dysfunction, and serves as a great template for us cell biologists to try and model human aging in vitro, where we can manipulate DNA damage, oxidative stress, autophagy, and senescence. And this is what we've gone on to do in this series of studies here. So this is looking at oxidative stress in vitro. So these graphs at the top, again, the different types of bisphosphonates being exposed to hydrogen peroxide. So the little white bar on the left of each of them is the control, whereas if you expose them to hydrogen peroxide, you get a big increase in cell death. But when, again, these are exposed to low doses, picomolar doses of bisphosphonate, this reduces the amount and protects against this level of cell death induced by oxidative stress. In some cases, it's 50%, but in other cases, as you can see from some of these heat maps in the THP1 monocyte macrophage cell line, a picomole dose of alendronate actually prevents entirely the level of cell death that would be induced by oxidative stress. Now, senescence, of course, is another big mechanism when it comes to aging, and of course, Lynn leads the field in senescence work, and specifically here in Oxford, and we'll be talking a little bit later in the meeting. This is an interesting process where cells get ejected from the cell cycle as they age, but stick around. They don't proliferate, but they don't die either. They take on a bloated appearance, but and still are able to spew out a toxic cocktail of cytokines and chemokines, which impact that local and possibly systemic environment. So what we were able to do was to mimic this in vitro by inducing senescence, by inducing DNA damage. So what you can see in the images on the left, in the control images, we just have the placebo for zoledronate, and we get a lot of staining for this beta-gal, which is associated with senescent cells, but zoledronate treatment is able to protect against that. In the other bisphosphonates, which are shown in the bar graph, in the control on the right, you can see that that's the sort of level of cell death that the percentage beta-gal positive cells that you'd expect to see, senescent cells that you'd expect to see when you induce with mitomycin, as we've done. But addition of almost all of these bisphosphonates is able to reduce senescence. And this is shown using this foci staining across the bottom as well. And Graham's already mentioned this, so I won't dwell on this for too long, but his work with Ilaria Bell and Truno up in Sheffield was able to show inducing DNA damage by irradiation in vitro and in vivo also was able to be protected by the use of zoledronate. Now I'm skipping now to one quick heat map from our human proteome screen, which I'll say a little bit about in a minute, but I just wanted to just show you this because it really indicates how, if you look at this SAS, which I mentioned, of course, is a large cocktail of different cytokines and chemokines, which are spat out by senescent cells, we can see an overall decrease in expression as we go from the 0-month treatment of zoledronate to the 18-month, and then again another decrease over to 36 months in patients who have had bisphosphonate treatment. What I'm looking at here is that change from red on the left to slightly bluer and then much bluer as we go across. And this is just taken out the top 20 or so of these SAS to highlight the fact that a number of these factors which are downregulated in patients who have received zoledronate are factors that we would associate with changes in inflammatory status, such as a significant reduction in transcription factor NF-kappa B, as well as a reduction in IL-6. So again, as I mentioned, this was a human study that was done looking at zoledronate, and samples were collected at baseline, at 18 months, and again at 36 months. And where our bioinformatic friends have been doing analysis on this large volume of data that we've got, some of which has only just been generated in the past couple of days, but which I wanted to share with you, is showing that we see about 1,000 proteins that are changed in patients who were differentially expressed from those who have received bisphosphonate from before they received bisphosphonate. So we have 1,000 going up and we have 1,000 going down. And again, this is another heat map showing the top 50 differentially expressed proteins in a column running down the right there. So we're at the stage now where we're aiming to try and pick through this list of interesting proteins and try and match them up to see with what might be happening with the number of these aging-related disorders, which we know bisphosphonates are likely to be protective against. But what we've been able to do up to this point is to do a pathway enrichment analysis where we're not really looking at individual proteins, but we're able to cluster them related to proteins related to the specific biological processes, proteins related to specific cellular components, or a molecular function. And this has told us some really exciting data so far, where a number of the highly regulated proteins that I've picked out here, that you might be able to see with my cursor, highlighted in red, are proteins associated with oxidative stress. These are the ones indicated here in the red bar, which is pretty much what we would expect to see. It tallies with what we've seen in our in vitro studies, that bisphosphonates are protected against oxidative stress. In green, we've got activations of T cells, which might fit with what we see with the gamma delta T cells. On the right, we've got in dark blue these protein groups associated with small GTPases and RAS proteins, which is what we know bisphosphonates do and inhibit the formation of these. So this is acting as a very good internal control for us. But also in amber,
but you wrap it up.
Yeah, we're almost finished, David. Thank you. So in amber, we've got autophagy-related protein clusters, which are highlighted, and surprisingly, which I really wasn't expecting to see, is that in turquoise here we've got protein groups which are associated with telomere maintenance, positive telomerase activity, and regulation of telomere capping, something that we haven't come across before, that bisphosphonate groups might actually play a role in telomere function. And the final surprise to us was that there was this group of proteins that is associated with the sensory perception of sound, and this is mixed in on an expression level associated with, compared to the skeletal system or collagen breakdown products, which we know bisphosphonates would have an effect on bone. But actually, it ties in really well with the study that came out just about 10 months ago, which showed that bisphosphonates are also able to protect against hearing loss in a mouse model of hearing damage, where treatment with zoledronate was able to restore ganglion neurons between synapses. And this was likely mediated by the inhibition of the mevalonic pathway. So we're following this up now with our colleague Mike Bowl at the UCL Ear Institute. So in summary then, I think what we know bisphosphonates induce apoptosis at high doses, but what we're really starting to show is that at low doses, and sometimes at very low doses, we actually get a stimulation of cell growth with bisphosphonates, a protection against oxidative stress and DNA damage and senescence and things which we might expect to see in an aging environment, as well as this activation of T cells, which may play an immunosurveillance and an antiviral role. And our human secretome data is going to keep us going for quite a while yet, but has already shown that we see changes in the senescence-associated secretory phenotype, as well as changes in protein clusters that we think are going to be really important in how aging is managed. So this is my final slide. And I just wanted to come back to the title of the session, which was how do we build on the potential for bisphosphonates. Graham's highlighted how a number of clinical observational studies have shown that bisphosphonate use does lead to beneficial effects in a number of aging-related disorders. And now we're starting to show mechanisms through which these might be mediated. We've got a lot of preliminary data that we hope to try and take forward. We just need a bold and generous funder to try and help us to do that, because bisphosphonates might not just be useful for individual disorders as well, of course. We're showing one agent that, as Graham described at the beginning, is very cheap and cheerful and has the potential to impact a number of multimorbidities. So I'd just like to say thank you to my team and to Helen and Jinsen who did all of this work, and of course for Graham for getting us involved with bisphosphonates in the first place. Thank you, David. I'm sorry if we've run over.
Thank you very much, James and Graham. They were really insightful talks there, and it was really interesting to hear the story of how we often talk about bench to bedside. Well, this seems like a story of bedside to bench, where we had clinical observations first and then went back to study the mechanisms. There's lots of really exciting questions coming in the chat, and I would encourage people to continue posting questions for Graham and James there. We're unfortunately really short on time, so we're not going to have time to take any questions on camera. But if members of the audience continue to post their questions in the chat, I'm sure if I ask James and Graham nicely, they'll be able to go into the chat and answer those. So thank you very much, everybody. We're now going to take a very short break, so please grab a glass of water, have a little walk around, and we'll be back at 2:45 for a series of 3 presentations. So thank you very much, James and Graham, and thank you very much to the audience for your attention.
Thank you.